Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. Welcome back to another Tools for Looking into the Brain episode, our series in collaboration with Scientifica. And so far in the series, we've had a couple of episodes focused on neuroscience techniques, from electrophysiology to optical imaging, and we've had a couple of episodes focused on research journeys and advice for early career researchers. And today, we hope to get a bit of both, as we are joined by Dr. Silvana Valtcheva, a principal investigator at the Max Planck Institute for Metabolism Research in Cologne, Germany, who started her lab in 2022. Her lab investigates the neural circuits and plasticity mechanisms shaping sensory perception and hormonal release in the postpartum brain to drive behavior. Prior to this, Silvana earned a bachelor's degree in biology and a master's degree in neuroscience before pursuing a PhD in activity-dependent synaptic plasticity, the role of astrocytes, and developmental changes in the control of plasticity with Professor Laurent Venance in Paris. Following this, Silvana joined the lab of Professor Robert Froemke in New York City as a postdoc, where she investigated the neural pathways and synaptic mechanisms important for the detection of offspring vocalisations in maternal mice. Reading some of Silvana's previous research, I was extremely impressed with the wide range of techniques used to uncover the neural circuits linked to behavior, and it's therefore not surprising that her lab currently also uses multiple state-of-the-art approaches in mice to understand the postpartum brain. And today we will be talking about Silvana's research journey so far, and hopefully hearing a little bit about her ongoing work to uncover the neural circuitry of maternal behavior. Welcome to the podcast, Silvana.
Thank you, Clara and Carolina, for this super nice introduction. Very complete.
Very impressive. I'm so excited to dig into your journey.
Yeah, and why don't we get started by you telling us a bit about your journey and how you got to where you are now?
Of course. So my journey, I'm passionate about synaptic plasticity since forever, since even before I started my grad school. And here's just a fun fact. I actually went to the university to study pharmacology, and I did a lot of chemistry, but then I also had one neuro course where I actually learned about the Hebbian theory. And then I thought it was absolutely wild that, depending on which neuron fire first or the sequence of this firing, then long-term plasticity can be potentiation or depression. And I think that this is completely insane. And then at some point, I actually realized that I'd rather go to neuro than pharmacology, because I can still use neuropharmacology tools to really track and dissect different plasticity mechanisms. So the only thing I wanted to do in grad school was to study any sort of synaptic plasticity. I actually didn't care about brain area. I wanted to do spike timing dependent plasticity because it's a Hebbian learning rule. But really, for me, the system didn't really matter. And I wanted to learn how to patch because I was absolutely fascinated about single cell activity and how you can really see at the single cell different channels participating in synaptic learning rules and memory at a neuronal level. So that's what I did during my PhD. I basically just patched all over the place, mainly in striatum. I did some monkey patch in cortical slices from monkeys. So that was that. I was really only doing spike timing dependent plasticity, very cellular based. But then during my PhD, I kind of started wondering, obviously, how experience shapes these synaptic learning rules, and starting to read about how different trainings and learnings in adult animals are kind of really underlying emergence of new behaviors and how synaptic plasticity is shaping that. And so what I noticed is that obviously something that we know already, that in adulthood, humans, but also other mammals, they need a lot of training to acquire new behaviors and actually to learn how to associate a given sensory cue with a new behavior and to kind of like make sense of the perception that they're having from different sensory modalities. But also what I noticed is that in a very particular model of adult plasticity, so this is the postpartum period, so I found out a lot of old papers basically claiming that there is a tremendous amount of plasticity in the postpartum brain, and it's kind of a switch, and new behaviors emerge all of a sudden. So we all know about so-called a maternal instinct, which is actually a complete myth. But for me, it was curious that we as adults need to train a lot. So if you want to learn a new language as an adult, you need a lot of courses. And you would never speak it as good as if that was your second mother tongue as a kid. So your window of opportunity to learn new things is really, really closed. But then I found out, okay, so maybe the postpartum period is kind of like a new window that's opening where drastic amounts of plasticity actually happen to really prime the brain to be able to acquire new knowledge about sensory modalities, which are completely new. So we can imagine all of the olfactory, tactile, and auditory cues from infants that are kind of bombarding all of a sudden a maternal mammal. And so the mom kind of seems that she instantly knows what to do. And I was like, oh, fantastic. Like, how did she learn that? I mean, the non-maternal mammals, the virgin females or the dads, they need more training, but she's like super expert. This is crazy. Is it a switch? What is going on? And so I actually decided that I really want to dig further into that. So that's when I joined Rob Froemke's lab at NYU to really study plasticities in the maternal brain. So Rob's lab, has like multiple different lines of research focused really on sensory perception and neuromodulation. So my project focused on oxytocinergic system and its involvement in the perception of infant cries, in this case in mice are vocalisations from pups in the maternal brain. And so that's where the whole journey started. So currently in my lab, we like to think of the postpartum brain as a framework to really study different layers of plasticities. Kind of this, as I said, new window of opportunity where animals can learn new things. What the field started understanding for the past couple of years is that there's no such a thing as, just like switch in behavior, but actually moms still need some training to start really excel at parenthood and taking care of the pups, but it's actually much faster. So for us, what is curious is really to dissect the individual contributions of hormonal switches that happen during pregnancy and delivery with actual experience-dependent plasticity mechanisms that occur while the mom is interacting with pups over time. And so something interesting in mice is that the postpartum period is very well defined. It's only 3 weeks because they will lactate and nurse the pups only during these 3 weeks. So it's a very defined window that we can capture. We can capture the before, after, and we can actually look also at neuroplasticity during these 3 weeks. Because previous research had pointed out that actually behavior and hormonal responses, they're different during these 3 weeks, which actually points out that it's not a single static state. It is very dynamic, and that all of these mother-infant interactions, they obviously adapt over time. In humans, the postpartum period is not a defined window. The postpartum period can be many things, and it's not defined just by breastfeeding the infant. A lot of women do not breastfeed, and that doesn't mean they're not in the postpartum period, right? So it's very debatable. What is a postpartum period in humans? Is it a couple of months? Is it 2 years? Is it 3 years? What is going on? Is it 18? Is it when the kid actually leaves home? So it's very debatable. So in rodents, actually, this is a fantastic system where we can look also at the aftermath of the postpartum. And so in the wild, mice live in colonies. And so once the pups are weaned, they would actually re-enter estrous cycle. So they'll start ovulating again, and then they'll breed again, and then multiple times per year, they'll actually have litters. In laboratory conditions, what is great is that we can isolate them artificially. We prevent them from breeding again. And so we can study so-called ex-moms or weaned moms. And actually, we can ask very concrete questions. We can ask, what are the aspects of the plasticities that we observed during the postpartum period, which actually go back to so-called virgin state, because the animal should be, ready to breed again. But also at the same time, human and animal research have shown that with subsequent litters and with subsequent kids in humans, parents gain, or moms, they gain so-called parental capacity. So actually they're quicker in understanding what the baby needs. They're just quicker to respond. They're better. They're more confident. And so in mice is the same. They actually would have bigger litters. They would be just better at performing a lot of the parental behaviors, which means that some aspects actually should be long-lasting. So we might be having plasticities that are reversible and some that are long-lasting in time. And obviously we can compare these lactating or postpartum mice with the ex-moms, the weaned moms, and at the same time to a third cohort, which are the age-matched virgins, which have never bred, never mated. And this is, again, an artificial thing we do in the lab, because in the wild, actually, the biggest percentage of the mice will go through motherhood, will actually breed. And this is a rodent-specific thing, because in other species, for example, hyenas or meerkats, only very high-ranking females will be breeding. So interestingly, in the wild, being a mom is not a given. So actually not every female can access to this, which also brings us to something, interesting to think about, whether this is a very particular life trajectory in adulthood for specific individuals. Again, this is not for mice, but just conceptually thinking about mammals, that being a mom is not the norm in a lot of mammalian societies. And also we can actually look at phenomena that are sex dependent. So these occur only in females and only in a subset of females. So something very specific. So we like to think about the postpartum period as really an equivalent of the early critical period during early development, but in adulthood. So it's really this window that opens, and then animals have the possibility to very quickly acquire new skills and to make sense of new sensory modalities.
Yeah. I just wanted to say two things that came to mind while you're speaking. First of all, I found it really interesting how you basically described what we may view as innate as just being super fast learning, because I've never thought about it that way. But also even in the early critical periods, things that animals do, we would think is innate. Maybe it's also just the ability to learn really quickly. And the second thing, I just wanted to say that it was really nice to hear your story, because I found so many parallels with my own life. And my first interest was also synaptic plasticity. And the first thing that made me want to study neuroscience was watching a video about Hebbian plasticity. And my first neuroscience technique that I just needed to learn was patching. And now I'm also finding it more interesting how to apply plasticity to more naturalistic settings. So hearing about your journey for me personally was very inspiring.
That is fantastic. I have a funny quote from myself that everybody makes fun of, but it's that I once said that every paper needs to have a slice figure.
Yeah.
That's the magnifying glass of anything.
Yeah, I wanted to ask a few clarifying questions. It's so interesting. I really enjoyed the way you painted the picture of pregnancy and the changes that can occur to the brain, as well as the different social hierarchies and how it's not a given to be a mom in the wild. But in terms of the plasticity, so just to clarify, is it plasticity only during the postpartum period, or is it during the gestation period, during the pregnancy? And also, is this specific to one brain region, or is it across the brain? And also, does this plasticity relate to either innate or more maternal behaviors, or is it more just now you have all this extra plasticity, go learn French if you haven't?
million-dollar question.
Yeah.
I'll tell you what we do in our lab first, and then I'll point out to other labs doing it kind of a little different. And I think we're all looking at the same problem from different perspectives. And that's where the community is very important and really try to tackle things. In my lab, for the moment, at least, again, the lab is 2.5 years old. But what we're doing is to compare first virgin mice with postpartum mice in different postpartum weeks. And then if we find differences, we're trying to track back and to understand exactly, is it during pregnancy? Is it during delivery? Is it at the very, very, very first day and the first encounters with the pups? So if you remove the pups immediately after delivery, what is going to happen? If you remove them second day, things like that. So we're kind of tracking back, because this is the way to actually tackle, as you said, things that are happening during or before the postpartum period, things that depend on the presence of the pups, or they actually have been triggered by the hormonal changes in the mom. There's excellent research, for example, from Johnny Cole's lab. Probably you're familiar with him. He's in London at the Crick. So his lab are doing extremely important work, actually looking throughout pregnancy. So they would be starting from, virgins throughout pregnancy, and then checking in early postpartum. And so it seems that exactly there's stuff happening during pregnancies. And so here is the whole conceptual framework that we're trying to reconcile with also other people's data, is that whether these hormonal changes during pregnancy actually serve as a kind of a scaffold for future very fast buildup of plasticities that are linked to experience. So as you were saying, this parallel with the critical period is just maybe it actually biases different brain networks to actually very quickly start acquiring. So you need minimum amount of training because evolutionally it's extremely important that this happens quickly. And you should actually remind me the rest of the question.
So whether this plasticity happens in a specific brain region or if it's brain-wide?
Yeah, so short answer, we do not know. Long answer, in the lab, at least we're looking in hypothalamus, thalamus, brainstem for the moment. We always take subcortical. Human studies have imaged the cortex and hippocampus extensively. The problem with human studies is that you can rarely recruit the same patients, obviously, over time. But they have shown some, very important data on that, some long-lasting changes. So there is a bioarchive study pointing out to the proteome of different brain regions, actually during the postpartum and afterwards. And so at least at the level of proteins, it seems that in the hypothalamus, the proteome profile, it's actually after weaning kind of goes back to like virgin state, which you can think it makes sense because this is, a core center for, reproductive behavior. But then the proteome of prefrontal cortex has a different profile. So we can think about that maybe the learned skills actually are transferred to different networks for like long-term storage, while networks involved in reproduction and, a lot of innate behaviors, they're more on and off switches. I'm painting a cartoonish idea of that, but it's very interesting. So indeed, actually, the locus of induction of certain plasticity is not necessarily the locus of maintenance. And I believe this is also, human studies have shown that also on a network level, this is true. But yeah, we don't know.
Yeah. Well, you've kind of already answered, but when asking about whether, yeah, you have more plasticity and you said like it can serve as a kind of scaffold, and my question was whether this can only serve for maternal specific acquiring of knowledge, or if it's like, okay, you have this 6 week window, go learn a language if you haven't done so because your brain's extra plastic.
Yeah, so I wish during the postpartum period we could learn different languages and then, sign up for all the courses. Problem is that at least humans report a massive amount of brain fog, stimulation, and exhaustion. So that might make sense because then you're ultra hyper focused on what is very important, and then you save your energy and resources for caring for the infant. So that being said, but this is just my own hypothesis, I kind of believe that skills that you have acquired because you had to care for the infant as a mammal can actually be applicable later in life. So think about, a mammalian mom in the wild. She needs to find the best place to hide the babies, to check food sources, to be extra vigilant about predators, and just in general know her environment very well, and maybe take strategic decisions that she wouldn't be taken otherwise if the kids were not around. And then I can imagine that later on, these actually skills are maintained, and then animals are much better performing in general. Maybe they also gain, social capacities. Maybe after experiencing having a teenager, you're actually very good at negotiating, or you're super resilient to frustration after you've been having a toddler. I have no idea, but this is something interesting to think about. And I always think about elephant grandmas. They always know where to bring the herd to find water and food sources, and they have this crazy memory. And it could be possible because, elderly females, they had to take care not only about themselves, but probably during their lifetime of their own offspring. And also a lot of females in different mammalian species, they co-parent, they take care of other females' kids. So maybe you're very right that actually these skills are transferable, but not immediately during the postpartum. Maybe later in life, you're like, oh, I had a kid. It was so difficult, and afterwards I can build a lab. Nothing can scare me. I can imagine things like that, but this is my own opinion.
Yeah.
And in mice, this can be really transferred very concretely in the lab. So you have postpartum moms, you screen them for certain behaviors. Then after weaning, you can actually test their social interactions, their stress resilience, and these kind of things.
That sort of leads me to my question, which is a basic one, but what sort of maternal behaviors do you actually look at? Because I know in your previous work you've looked at like pup vocalizations and responses to that, but do you also look at other maternal behaviors?
Yes, so we studied two things at the level of mom-infant interaction. So the first one is how sensitive the moms and how reactive the moms are to the infant cues. You mentioned already the auditory cues. We're also looking at tactile cues. Later in olfactory, but we're not there yet. We believe they're a little more complex, so we need to understand some other things first. So this is basically how quickly the mom will react after hearing a pup or having a pup attached to nurse and things like that. So this reaction can be a behavior which is towards the pup. For example, pup is isolated and the mom hears it, runs towards it, pick it up, brings it back to the nest. But it can be not directly pup centered. So for example, there are other vocalizations of pups, which they emit them not when they're isolated from the nest, but when they're inside the nest. So actually, when they start vocalizing, the mom will be turning in the nest, switching position. So technically, she's not directly engaging with the pup, but by changing position, the pups might be gaining more access to the nipples to nurse, for example. So yeah, it's subtle difference, but it can be maternal behavior without the moms directly handling the pups. Let's call it that way. And then the tactile cues, obviously they come during nursing, right? When the pups are attached to the ventrum, to the nipples. But tactile cues also come from the mom handling the pup and licking it and grooming it. So these are cues, information processed from the orofacial region of the mom. So the very concrete behaviors that we're looking at are obviously the mom exiting the nest, picking on the pup, the mom turning in the nest after hearing the pup, the nursing, obviously, so there are different nursing positions and things like that, and how much time the mom spends in the nest with the pups, and just being engaged with the pups versus doing stuff on her own, right? Because she also needs some me time, rarely, but it happens. And then when she handles the pup, what exactly is happening? Is she licking, grooming? Where is she handling the pup the same way as she handles a food pellet? And things that actually haven't been looked before at this like very fine grain level.
Mm. Yeah, that's interesting. And I was also wondering, from my own experience, I know that different mouse strains have very different maternal behaviors, and certain strains are like much more maternal, so to speak. And I was wondering if you ever look at this variation, but also the variation in terms of like how the size of the litter alters these maternal behaviors, because it just made me think if you have like many more pups to deal with, then does the mother get more stressed out, or do they actually cope better? Yeah.
Yeah, so we haven't looked at other strains. That would be fantastic. We believe we first need to check what's going on in the current strain, like the Black 6 mouse, to have like a clear phenotype that we want to compare to others.
Yeah.
And then, so one versus many is a very tricky question also for humans, right? You have a baby crying, at the same time you have a toddler, and then you have a preschooler, and then they all need different things at the same time. What for mouse moms haven't been answered yet, and we're trying to understand what's going on, is how come she will leave sometimes 6 to 8 to 10 pups in the nest unattended while she go fetches one crying pup. And so we're really looking also at this multisensory competition between the suckling cues, the tactile suckling cues, and the auditory cues, and whether they can converge in the same region, and how actually the sensory information is competing, so you can prioritize one than the other, and whether the context also would matter. And again, I come back to the wild, right? If there's a predator around, would the mom leave the nest? And people have shown that actually, depending on the age of the pups, she would either stay and defend them or leave and run. And you can think about evolutionary, it might make sense. If the pups are a little older, the decision here is that, probably they're able to take care of themselves. They might be able to run. And for the mom, it might be better to actually run and abandon them and re-enter a new breeding cycle. So yeah, I think it's all about optimizing resources. But yeah, we're at the very early stages, just understanding how the sensory cues are competing with each other. And again, as you were saying, if there are no pups in the nest, whether for the mom is easier, maybe the latency will be shorter, maybe she'll hesitate less to go out. Yeah, excellent question.
Yeah, there's so much to find out still. So I wanted to switch gears a little bit and go back to your research journey. So one thing I noticed is, as you explained, you were very focused during your PhD, topic-wise and technique-wise. And I noticed that your postdoc work involved many, many different techniques. I was very impressed, from like patching to chemogenetics and in vivo manipulations and behavioral analysis. And I'm assuming that those sorts of things are also applied to your lab's work nowadays based on the research that you've told us about that's ongoing. And so I was just wondering, how did you find making that transition and, learning all of these new techniques and gaining the confidence to the point where you can also start your own lab and set them up yourself? Yeah.
Yeah, you're very right that actually that was my probably number one goal during my postdoc, to learn as many tools as I can, not just random tools, but the tools that I believe will be useful for me in my lab. to tackle different questions of interest. And it was absolutely with a very clear purpose that I wanted to acquire X tools. I'm just going to add here that I believe that during a certain career stage, you should always be already looking to the next one. That being said, as a PhD student, you should already be preparing yourself for a postdoc if you want to do one. And as a postdoc, you right away start preparing yourself for a PI potential job if that's what you want to do. It's not that first year of grad school gonna think about your postdoc, but very early on, you need to start preparing, because neither PhD, neither postdoc are long-term careers. They're stepping stones to something else. PI is one of the job opportunities you can do. So that's really my suggestion. So during grad school, I was like, oh, it's 21st century, and I know this one single technique, which is amazing. And it can be applied to anything, but still I need to really expand my toolkit. So that was an absolute realization. So I had probably like two main goals. One is to be able to record activity in vivo in an awake animal from multiple neurons and from a single neuron. So that's when really I decided I want to do patch in vivo, as well as in my postdoc, at least, I did fiber photometry, which is recording calcium signals from a population of cells. This is one. And the second is that I wanted to be able to perturb neuroactivity, which is where I did DREADS and OPTO, actually, exactly. And I also wanted to be able to do some anatomy. So I want to say that I did my PhD in rats, actually. So the tools at the time in rats were not as developed as now. Now we have transgenic lines and viruses that work in rats. It was really not the case before. So for me, switching to mouse actually gave me the opportunity also to do a bunch of viral tracings and real anatomy and dissecting of neural circuits and things like that. So that was absolutely intentional in a way. It was not really learning random techniques just to have many on my CV, but really techniques that I think I would benefit later on as well.
Yeah, of course. It's also very fitting given that the field of like neural circuits and behavior is just including so many new techniques nowadays as well, like all the different sensors and types of imaging and electrophysiology.
And they're all transferable across projects, actually. So that's the good thing. In my lab, we probably have like a single technique, which is only for postpartum mice, which is detection of suckling activity and milk letdown. But all the other techniques are transferable across any project. And about the learning, with the risk to sound cynical, but anyone who can patch can learn any other technique. So
this is the exact same message we got from Elisa Galliano, who we did another Scientifica episode with. It's nice to hear that.
It's really interesting to hear you say that when you start your PhD or you postdoc to be looking ahead to the next position. We've had many different PIs talk about their journey and that how, like, with them it involved a little bit of luck, a little bit of serendipity. So it's interesting to, I guess, hear a different perspective. And I think that many people don't think about what they're going to do next until the very last minute. Yeah, can you tell us a little bit about your thought process in that, how it's actually to your advantage, and also the issue with timings in every single job because you time out of opportunities once you finish your PhD?
Yeah, totally. So I think for me, serendipity plays an enormous role. So I do believe in opportunities, in luck, there's a lot of noise in the system. This is fine. This is something we cannot control. And what we can control to some extent is how we deal with certain situations. And I always like to think about it's like surfing. So you cannot control the sea. Sometimes you have big waves and small waves, and the wind is different. But if you can surf and there is a good wave coming up, you're going to catch it. You know how. So it's a little bit like that. So I wanted to be prepared the best I can for when an opportunity comes. And obviously that's a proactive searching. I'm not like sitting in my office waiting for someone to offer me a job. But if there is a job opening or a funding opportunity deadline, I can go for it. So that's one thing. Second, I think you should know, at least to some extent, why you want to open a lab. Like a very clear why. Sometimes it's a personal why. Sometimes after you open a lab, you find many other reasons actually it's good for you or bad for you. But you need to have some answer to that question. And I think a very vicious circle could be that you are just jumping to the next level from PhD to postdoc to PI because you think this is just the only opportunity. I don't know, it's just one-way street and that you're not looking around. I think that can be deadly for your creativity and basically your happiness. So you need to really know why you want that. And as you mentioned, to be very aware of the deadlines, because exactly you're eligible for certain fundings, depending on how many years after your PhD diploma is. You need to be aware of different environments, different countries, and basically the whole community. You need to build your network extremely early. And when I say early, this starts as early as a grad student. So ideally, people will know you well before you start going on the job market. Mm-hmm. That's really a tip I want to give. And also get your priorities straight, actually. Because, you can be a PI in many different universities or institutes. You can have teaching or non-teaching, different countries, different cities. So really what you want from life and from your lab, ideally, and have contingency plans. Different types of PI jobs that are acceptable or not acceptable for you. And if ever you cannot find a PI job, what else you want to do? Because I believe, we're capable of many other things. I can speak for myself. I can be happy at other jobs as well. I'm not only happy being a PI. So there are many things that are super interesting to me where I can feel completely fine. And so for me, I had plan B and plan C. And so PI was one way of doing science and staying close to science, but this is not the only way. I think that's very important to have because it prevents you from being desperate sometimes, right? So things can become very dire if you think you have really only one option. And at the career stage of advanced postdoc, none of us has only one option. We're overly trained, extremely skillful, very creative. So you should not see a PI job as the only thing that you can do. I think you need to be also very realistic about the numbers, about the job openings within a single country, depending on the size of the country and the funding, landscape. Sometimes jobs are extremely scarce, actually, and you can count them on one hand sometimes in certain fields. So yeah, I think this clarity will take away a lot of the stress and the mystery around job application and really understanding what you can control and what you cannot. You cannot control job openings. You can control performing at an interview. That's it.
Yeah. Yeah. One thing that I don't know, I can only speak for myself personally, but even though academia is really appealing to me, I am also a little bit curious to try something outside of academia, even if it's just for a year or two, whether it's in I don't know, the startup world and industry, science communication, policy. There's so many cool things that you can do with a science background. But the sense that I have is that if you leave academia, either because you time out of things or because, you left academia, it's hard to get back in. So the way that I feel is I can't explore these other things that I may have an interest in because if I want to do a postdoc later on, I need to do that immediately after my PhD or else I won't get fellowships. Yeah.
I hear you 100%. So I cannot really speak about, the deadlines, for example, if that's going to affect your eligibility for certain grants. But I think, at least from my knowledge, few people come back, not because they cannot, because they don't want to. A lot of people around me have left academia either after PhD or after postdoc or after opening a lab and then closing it. And there's no interest of them coming back to academia because, again, there are many ways of doing meaningful science and staying close to science and contributing to knowledge without running a lab. I also know examples of people who, as you said, they've worked for a certain amount of time in the private sector and then they're back to academia. So I do believe the switch is possible. I think maybe the limiting factor would be these grant opportunities and the ticking clock after PhD defense. But yeah, I'm curious about actual, real statistics of people who left, and I put quotation marks because for no other job we say leave. I feel it's kind of a academia specific thing when someone just changes jobs and we say leave.
Yeah.
If they left the cult.
Yeah, exactly. That's what I was thinking.
Yeah, but I'm curious about actual statistics. Why people don't come back? Is it because they cannot? Otherwise, I assume they just don't want to. So obviously there's this ticking clock about the grant opportunities, but many labs have their own funding, right? So I know of postdocs who changed jobs for 2-3 years and then joined as a postdoc a lab, and the lab was paying them from their own budget, right, without grant application. What I want to say is that life is too short to get stuck into something. So if you feel exploring other opportunities, just do it. I don't believe it's irreversible.
Yeah, thank you so much for this. I think it's actually very refreshing to hear somebody who has actually made it successfully to PI young, that there are actually other options as well. Because, yeah, too often I hear people just act as if only hardworking people will make it and those are the creme de la creme.
Yeah.
And it's just not true. And I really like your attitude of do it because you want to. But it also makes me curious to ask you, like, when do you think you knew you wanted to become a PI, even if it wasn't your only option? And what is your why?
For sure, during grad school, I could already see myself as a PI. That being said, I always checked other opportunities because, exactly as Carolina mentioned, I didn't want to miss out on things. But my PhD, for sure, I knew, and I think a big contribution had Laurent Venance, my PhD supervisor, who was a fantastic mentor. And it's just seeing someone who comes every day and is extremely passionate about science and just going to everybody individually and just asking about experiments and just training students and being extremely involved and just be curious and having fun. And I thought it was just like the best job in the world. That's from my naive PhD perspective, but I like the idea to run your own show. I mean, it sounds crazy, but you do have your small company in a way. And yes, you have limitations, which are funding, 90% of the cases, but you kind of can almost do whatever you want, obviously within the legal requirements of animal experiments, but you can actually wake up with an experiment in mind or with a question and do something about it. And I found this, it's kind of a pseudo freedom, but it is freedom in a way. Yeah, extremely appealing. And I loved the idea of interacting with different people. And I love the idea to not be the only brain. And so that's exactly what I tell my people. I love being wrong. Tell me I'm wrong. Prove me the opposite. Challenge me. Let's have that discussion. I don't like to be the only person deciding, or I'm not running a one-person show. I was imagining like a huge ship where everybody have something to do and just this whole machine is working because everybody have their own tasks and their own contributions and they see things with a different angle. And we can really address very complex questions together, actually. Like really upgrading beyond having your own project as a PhD student. Yeah.
Yeah, I think maybe that was your answer, but is that your why? Because you like the job and what it entails.
Yeah, I mean, so I applied for a PI job because I had a bunch of questions in mind, which for the last 8 years, no one has solved. And I thought that someone needs to do the job. Yeah, real curiosity. But now, after running a lab, there are a lot of other things that I discovered that are extremely rewarding, which I didn't know before.
That's so good to hear.
Yeah. And I really also like the attitude of, applying for a PI position because you're passionate about it, but it's not do or die. And like a lot of PIs that I've spoken to and that we know, like that's very much the attitude. And I think it also puts you at an advantage when you're doing talks and interviews because you're not desperate. It's not your only option. And you can really advocate for yourself more if you have multiple interests.
I also think, relatedly, that your mentors in your journey make a huge difference because, like you, I am in my local environment surrounded by passionate, curious people, and that makes a huge difference because it's actually rare, I think, a lot of the time in neuroscience. And to maintain that mindset so far has helped me already when I struggle because I remember what's important. And yeah, I hope to keep that like you.
No, absolutely. I think the important is to exaggerate, but if I don't have fun, I leave tomorrow.
I
I'm
like
going
that.
to my PhD students to graduate, however. But I think it's really important to keep in mind we have the privilege to have options. We have the privilege to choose our job. This is rare. Few people can choose their jobs. So I think having that privilege, we need to make use of it, right? And of course, what we do for work is coupled to our ego. We cannot fully decouple it, but I think it's important to decouple as much as we can. And this is something I learned the first couple of days, patching, like the massive amount of frustration. And then I leave the lab and I think I'm a failure. And then you realize that has nothing to do with you. Like your experiment has nothing to do with you. It's your skill and that's it. And then you leave, whatever happens in the lab happens in the lab. You carry the curiosity about science outside of the lab, but your ego, you really leave at home. It is the same when applying for jobs. You're going to fail a lot of interviews. You fail grants constantly. That's normal. And yeah, you should just not get devastated by that every single time. And exactly just finding value in other things and knowing you're good at a lot of other things, I think it also helps.
Yeah, great message.
Uh, Before we wrap up, any final bits of advice?
I think maybe a few things as an advice as people who want to go for the PI career. I mean, I already mentioned that you need to know why you want to be a PI and build your network early and be realistic. But maybe the last thing that I want to mention is really how to distinguish yourself from mainly your postdoc supervisor and how to build your own brand. So that's important. And what makes your concepts and your research unique, actually, and how you branch off from your PhD or postdoc career. That's very important.
Be original.
Yeah, that's a really good message.
Yeah, thank you so much for joining us today and yeah, sharing your journey and your perspectives and your science. It's been a pleasure chatting to you today.
Yes.
Thank you all for having me. Yeah, it was super fun.
Yeah, it was really refreshing speaking with you, not only learning all about the science that you do, but also your attitude to academia, which I think is really great to hear.
And looking forward to seeing what comes from your lab in the near future as well.
Thank you.
Thank you for listening to today's episode, another one in our Tools for Looking into the Brain series with Scientifica. We hope you enjoyed it and do check out our website for more information about all of our episodes. Also, if you enjoyed the episode, share with your friends and family and follow us on social media at neuroverse_pod.